Well, welcome everybody. Thank you for joining us for our 2020 Wildlife Health Workshop. You can see the format's a little bit different. We're not able to get around to visit you in each of the regions this year. So we're going to give this a try. It's our first one, so we'll see how it goes. But for those of you who don't know me, I'm Krysten Schuler. I'm a wildlife disease ecologist at Cornell, and I work with Kevin Hynes and others on the New York State Wildlife Health Program. So we'll let Kevin introduce himself.
Hi, everybody. Can you see my face? I just see Kristen's face. Am I muted?
No, you're good, Kevin.
Oh, okay. I'm happy to join you virtually, and I think this is going to be pretty awesome. I'm glad you all were able to join, and I think you're in for a really good hour and a half or so of enjoyment and education.
And I'm Brenda Hanley. I'm a postdoc in the Cornell Wildlife Health Lab, and my specialty is mathematics, statistics, and computational biology.
Great. So what we're going to do today is I'll start off and talk a little bit about COVID-19. I know you all have heard a lot about it, but I'm going to try to just focus on the animal side of things and sort of what we need to know as wildlife professionals. And then Kevin is going to do a presentation on PPE and disinfection, what he normally does when we go around and do the health workshops, but just update it a little bit. And then after he's finished, we'll have Brenda talk about some of the modeling that's being done around COVID and how we can know certain things that we know. So this presentation is being recorded. So if you aren't able to stick around for the whole thing, you can always come back and watch it later. Or for those who weren't able to join us now, encourage them to watch it later on. And we'll have it up on the Cornell Wildlife Health Lab website in about a week. And we'll have some additional resources along with the presentation. So if you have any questions or anything, please go there. So I will go ahead and share my screen. All right. So like I said, we're talking specifically about how COVID applies to wildlife in this situation. But just as a bit of background, I thought it was worth showing this map again. And hopefully this map looks really familiar to a lot of you, because I think we've shown it at almost every single wildlife health workshop that we've done thus far. And we always point out that the Northeast is a real hotspot for emerging diseases. And I think it's relevant now to point out where some of the COVID situation is the worst, and that it really does, this map is actually really held up as far as where these hotspots are occurring. And a lot of that is related to human travel and trade and human density as well. So you've seen this slide before, just as a reminder that when we're talking about wildlife health, we always try to think about it in the context of One Health, which is a collaborative approach to health at multiple levels. So we're thinking at it from globally all the way down to the local level, and recognizing that the people and animals, domestic animals, wildlife, plants, and the environment are all shared. And it's really important to communicate these different factors. And I think the people are really taking that to heart now, especially with COVID. The other part that's important to recognize is that you need the right conditions for a disease to take off. So you can't just mix the agent and host, but the environment generally has to be right for that. So there's been a lot of talk about what kind of conditions does COVID persist in, and so that's an important consideration. We've also shown this infographic before. This comes from the World Organization for Animal Health, or OIE. So they have been reporting for a number of years these statistics about how 60% of existing human diseases are zoonotic, meaning that they came from animals. When we talk about diseases going from humans to animals, that's often referred to as reverse zoonoses. So a lot of these emerging diseases that we're talking about, like COVID, have an animal origin. And I think one of the things that's kind of gotten lost in this is that 3 out of the 5 new human diseases that emerge every year have an animal origin. So this situation that we're in right now is unusual because of the scope of it, but as far as diseases emerging from animals, it's not really that uncommon. So when we start talking about SARS and COVID, what we really need to be thinking about is what information can we gain from what we've seen in the past? So with this outbreak, the first cases were identified in what was called a wet market in Wuhan, China. And I'm not exactly sure of the origin of wet market, but basically it's a live animal market. So a lot of the markets in other countries operate differently, where they don't have the refrigeration and sanitation. So people actually want to see the animal alive to know that it's fresh, and then the animal is killed right there, which means that a lot of animals are mixing together with different species. You know, they're stressed from being caged and cramped, and so they may start shedding a lot more pathogens than they normally would when they're out in their regular habitat. So that allows for a lot of viral mixing. So right now, because we haven't seen this particular virus before, we're trying to understand the relationship to other coronaviruses. So the ones that are most noteworthy are the sudden acute respiratory syndrome, or SARS, that emerged in 2002. This is the one that's most closely related to the virus that we're dealing with right now. And we know that this virus went from a bat to a civet, which is a type of cat in Asia, and then infected humans. And there were just under 10,000 deaths associated with that. There was another one in 2012 called MERS, or Middle East Respiratory Syndrome, that went from a bat to a camel and also was able to infect humans. Now with SARS, that only happened once. With MERS, there's been a number of outbreaks associated with that, but there haven't been that many deaths. So the case fatality rate for MERS isn't very high. With the SARS-CoV-2 that we're dealing with now, which is the virus name, whereas the disease name is COVID-19, that virus they identified went from a horseshoe bat, and we still don't have the intermediate host, and then to humans. And there's been over 400,000 deaths globally thus far. But I think going back to that previous slide I showed you about emerging diseases, there was a study done by the EcoHealth Alliance looking at people in a province right at the border of Myanmar and China that demonstrated nearly 3% of the people there had antibodies for bat coronaviruses. So that tells us that there's been a lot of spillover from wildlife to humans in these viruses previously, but maybe they weren't infectious, or maybe they didn't have the right conditions to take off. And these coronaviruses are really challenging to study because they don't grow easily in cell culture. So they don't grow in a lab very well, which makes it challenging. But there are a number of human coronaviruses out there. I think they said there was like 4 of them that cause the common cold. So humans have been exposed to coronaviruses previously. And as I mentioned, horseshoe bats were implicated as the original source. And they have considerable genetic similarity in the viruses identified in this to the SARS-CoV-2, but not 100%. So we don't have a smoking gun, so to speak, for an exact match right now. So we know that there were multiple species of live animals available at this Wuhan market. You might have heard in the news that there were pangolins that were originally implicated as the intermediate host. Those have been kind of ruled out because of the genetic similarity issues. So the difference, I mentioned that Yunnan province to Wuhan, and that's a distance of over 800 miles. So we know that there's likely to be a lot of human movement of animals to these different markets. So animals are being moved around, and as I mentioned, they're stressed, they're being caged, and that could allow the virus to emerge. So here is a schematic of what the virus looks like, and it has four major proteins that we're looking at. The one that you probably heard the most about is this spike protein, and this is actually where the virus gets its name from, corona or crown, because there's a lot of spikes sticking out of this virus. And it's challenging to study these viruses based on the genetics because you can't always identify these different proteins exactly. So it's very complicated. But what we've seen thus far is with transmission, and we expect this to be the same for animals as well, is that you have direct transmission from human to human through respiratory droplets. It's possible there may be indirect transmission of people, the virus getting on something and you touch that and then you touch your face, but that doesn't seem to be as common. We are still trying to learn more about asymptomatic carriers and how they're shedding the virus. But one thing that I think is really interesting is that there is fecal shedding of the virus. And other coronaviruses in animals show that they not only get respiratory infections, but also enteric or gastrointestinal infections and can shed the virus in their feces. And some of the animal coronaviruses that are out there commonly cause issues with gastrointestinal signs, so things like diarrhea. Now, we think that in a lot of these human to animal transmission, there needs to be prolonged contact or high dosages for the animal to actually get infected. And some of the recommendations that researchers at Cornell and other places are coming out with is it might be really useful to study some of these animal coronaviruses when we're trying to understand the human disease. And this goes back to that One Health concept. So as far as cases in animals, I have a little asterisk here because this data is from the beginning of July, and so it may not be completely accurate right now. So there's been at least 17 cats and 5 dogs that have tested positive for SARS-CoV-2 worldwide. So not a high number, but we also have to think about the denominator of that number, and not a ton of animals are being tested. By the last report I heard, I think it was 200 and some animals had been tested in the US. And I'm sure you all saw that the first report in the US was from a tiger in the Bronx Zoo. It was infected via the zookeeper taking care of it. It also was able to transmit the virus to other big cats that became infected, and all of those animals have recovered. There were 2 domestic cats in New York that were some of the first domestic pets to be identified with the virus. There was recently a German Shepherd that was also identified in New York to be infected. This dog was reported in a COVID positive household, so it had really close contact with at least 2 people that were infected. And the other thing, going back to those, the fecal shedding part, is that this dog was also reported to have drank out of the toilet bowl, which could be another source of infection. One of the things that is troubling to me is our reports out of the Netherlands that 13 mink farms have become infected with COVID-19 in their animals that show both respiratory and gastrointestinal signs, and they've had an increased number of deaths. So they have identified that workers at those mink farms were the source of the virus, but they're relatively confident now that mink were able to pass the virus back to humans through close contact. And they're basing this on genetic work that they've done. There's also farm cats that have been on these facilities that have been antibody positive, suggesting that they've been exposed to the virus. So there's not a ton of lab research on these, and a lot of those have small numbers of animals. So these studies really didn't focus on whether animals could spread the infection to people. So we don't have a lot of information on that. But right now, and I'll talk about this later, we don't think that animals are a major source of infection. So ferrets, cats, and Syrian hamsters can be experimentally affected and spread it to other animals of the same species. Various species of non-human primates that are commonly used in laboratory research can get infected and become sick. Other common animals that we see, like mice, pigs, chickens, ducks, do not seem to become infected or spread the disease. And dogs may not become as easily infected as cats or ferrets. Now we're trying to figure out, and there's researchers working on this, there's a specific receptor called the ACE2 receptor in the respiratory tract, and they're trying to look at the genetic composition of those receptors to figure out which animals are most susceptible to the virus. And I saw a preprint of a publication, and I'm not sure if that's actually been published yet. And that's one of the challenges with this virus, is that so many publications are being put out there before they're actually peer reviewed, that you just need to take everything sort of with a little bit of caution about what the results actually are and whether it stands up to scientific rigor. So the CDC put out language that said, at this time, there is no evidence that animals play a significant role in spreading the virus that causes COVID-19. So the way that I'm thinking about it is that humans are actually the threat that we need to be thinking about to free-ranging wildlife populations. And there's pretty significant consequences to exposure to this virus. We could have wildlife mortality, which especially for some of these species that we're really concerned about, like bats that have already been hit hard by white nose syndrome, or other species that might be endangered or threatened. We obviously don't want disease to spread. We don't need a new wildlife disease reservoir. We already have rabies and things like that in some of these species, so we don't need to add another one. And when we talk about risks of potential spillover of the virus into wildlife, we're thinking about those activities that have sustained contact with live animals, where those animals would then be released and able to transmit the virus. So things like research or rehabilitation, education, exhibit, wildlife nuisance and control, captive rearing with the intent to release, translocation, and potentially trapping if it's a non-target species that might be released by that trapper. So the questions we're getting a lot are, when should we be testing animals? And right now, there's some interim guidance available. So going back to the World Organization for Animal Health, which is essentially the CDC for animals, their language said, sampling and testing of animals could be considered in situations where the tests will inform decision-making, animal case or population management, or public health response, or further the body of knowledge on transmission of the virus. And right now for DEC, any decisions on testing must be made in conjunction with the Departments of Agriculture and Markets and the Department of Health. So it's not just us going to the lab and saying, yes, we want the testing done. It has to be approved in advance. There has been some pushback of animal testing, that the resources, whether it's personal protective equipment or staff or reagents, should be prioritized for human testing. It seems like right now, the statewide testing it is at a level that people are generally happy with, so I don't think this is as much of an issue. But one of the concerns also is that communication of the test results should avoid inappropriate measures being taken against domestic or wild animals. At the beginning of this outbreak, a lot of veterinarians were really worried that people would start dumping pets because they were worried that they could get infected from their pets. And similarly, we don't want wild animals to have people threatening them. So we want to avoid a perceived risk of disease transmission from wildlife that would erode our conservation intention. So whenever we want to start testing, we need to make sure that we have a clear rationale for doing that, and that we plan ahead to whatever those test results are going to be. And whenever there is a positive test that we run at Cornell, we need to go ahead and send that for confirmation to the USDA, to their national lab as well, to back up that test result. So breaking that down a little bit further, the testing wildlife may be appropriate when we have a morbidity or mortality event, and we do that statewide, where we're looking for animals that might be sick or found dead without an obvious cause. So one of the things that we want to look for in this are any clinical signs or postmortem lesions that might be consistent with respiratory or GI signs. So, it's not coming out of the nose, diarrhea, things like that. And we also want to think about testing animals that might be found dead or euthanized when there's a suspicion of exposure to the virus as well. We can also consider testing when we're ensuring the health of threatened or endangered species or species of particular concern. Again, we want to make sure that we look for those clinical signs and that we have testing in place to prevent infection to these populations. And then the last one being for research purposes. So our preferred samples from a live animal are nasal or oropharyngeal or rectal swabs or fecal samples. When we have an animal in hand that is dead, we can do tissue collection through our necropsy. We need to make sure that anybody that might be thinking about this gets instructions in advance on proper sample collection, storage, and transport. Kevin is going to go over personal protective equipment when working with animals, and we can cover specific sample collection if there is a need to directly, and we've been in contact with people about this. We can help you coordinate sampling through New York Wildlife Health Program, and in some situations, testing may be required for a period of several weeks, so serial testing on the same animal to be sure that it's cleared the virus. You've probably heard that there are PCR tests and then there's antibody tests. PCR tests look for the RNA or the genetic material of the virus, and sometimes PCR tests can pick up RNA that's no longer infectious, so sort of dead RNA, so you get a positive test even when a person or animal isn't infected. The antibody tests are what we see after the infection has already passed, where the body has built up the antibodies for it. So some of the species that we're most concerned about are North American bats. There was a bat risk assessment done using little brown bats as the model and expert elicitation from about a dozen experts across the world. And what they concluded is there's a non-negligible risk of transmission of the virus from humans to bats. But that risk was reduced pretty substantially with the proper use of N95 respirators, dedicated clothing or Tyvek suits, and gloves. Now, just this past week, an infection trial was done at the USGS National Wildlife Health Center where they experimentally challenged big brown bats. They allowed those bats to exist over 21 days and sampled them consistently. None of the bats showed any clinical signs of infection, and they didn't identify any virus shedding in swabs, oral or rectal, during that time, but that study is not complete yet, and analysis of the postmortem tissues is still pending. Now, just yesterday, the Fish and Wildlife Service released some guidance saying that it is appropriate to handle bats if you are using PPE and following the CDC guidelines, and we'll be able to put that guidance up on the website if you're interested in looking at it. The next steps as far as infection identification, they're going to use little brown bat tissue cell cultures to see if those cells can replicate the virus. The other species that we're concerned about are mustelids, felids, and canids because of what we've seen in domestic animals. So there is guidance from the Association of Fish and Wildlife Agencies saying risk mitigation strategies include temporary suspension or postponement of field research and management involving direct contact between humans and animals. People need to be trained on the use of PPE and disinfectants, which is why we're here today. Or there's the potential to develop animal handling protocols to reduce transmission potential. We want to make sure that we design systems where animals are held in confinement, such as for exhibition or wildlife rehabilitation, to make sure that that reduces potential transmission, or if necessary, suspend those activities where feasible. And then making sure, just like you're probably talking about with coming back to work, that people are monitoring themselves and potentially getting testing. So what should wildlife professionals be doing? And of course, this is all what we know today, and it could be updated tomorrow. So it's good to keep yourself armed with the current information. Bats that have been taken into rehab in New York State are currently being held without a release date, but there are conversations in progress about testing both the wildlife rehabbers themselves and the bats. So we should have some updates on that over the next couple weeks. People engaging in Fisher and Martin research are working on an updated protocol for handling those animals following that guidance that I mentioned previously. Kevin Hynes issued a guidance document for wildlife rehabilitators for mitigating transmission of the virus. And then the interim guidance from AFWA. We would like those in the field and the regional staff to continue to monitor wildlife for mortality events. If you get calls from the public, we're interested in hearing about those. And just basically doing the same things that we've been doing, where we're collecting carcasses for diagnostic testing. What we're doing with those is that we have to rule out all other causes of death before we can go ahead and test for the virus. So we will be going through our normal necropsy and testing procedures. So that's all I have. I know it's frustrating. We've all been home for a long time, but hopefully it will be a situation that we can contain and make sure that we're not threatening our wildlife species with a new wildlife disease. So that's all I have. I'm going to turn it over to Kevin now.
Oh, Krysten, you have a question specific to your topic if you want to answer that.
All right, so somebody asked, does the virus need an intermediate host, or could humans derive it directly from bats? And I'm not entirely sure of the answer to that. Right now, the work that's been done to try to look at the virus in bats doesn't have exactly a match. So that's why they're thinking it didn't go directly from bats to humans, although there are a lot of viruses that can do that. So like Ebola can go directly from bats to humans. There's a lot of other viruses out there that bats are carrying. And I think we're just starting to learn about the diversity of that. So it's hard, I think, because there's the potential for so many different intermediate hosts based on where they identified the virus first. But that's something we're going to learn more about here in the future. So I appreciate the question. Thank you. Kevin, it's all you.
All right. Can everybody hear me? Can you see me? Is my face going to show up on this? Because I don't see it. I just see your face, Krysten. All right, so let's get started. I'll do this to Krysten. Personal protective equipment. What is PPE? PPE stands for personal protective equipment, and it's equipment that's designed to minimize the hazard of illness, infectious disease, or injury to you. So some good examples that I'm not going to talk about right today would be like a motorcycle helmet or ear protection. These earphones kind of remind me of ear protection that we might wear over at our incinerator building because that machine is so loud when we're over there. So it's equipment that's designed to minimize injury or illness. So the reason you wear personal protective equipment is to protect yourself from being sick or injured, but it's also to protect your coworkers, your family, your pets, and maybe your research animals if you're doing work with animals in the field. If you get some material on your hands and you transfer it to the motor vehicle that you're driving or to pens or other things that I'm going to talk about briefly when I look at the gloves, you could transfer those agents to one of your coworkers or bring them home to your family. So what are these agents? So the agents that we want to protect ourselves from are viruses, bacteria, fungi, prions, or chemicals or toxins. And some examples of these would certainly be rabies, right? So rabies is a viral disease that we deal with day in and day out. here at the DEC, and you guys have exposure to it as well. And it's a virus that is a zoonotic virus. So Krysten talked about these earlier. They're diseases that can go from animals into humans. And rabies is a zoonotic disease that can get into humans, and it could kill you if you do not get treatment in a timely fashion. There's also parasites that could go into you. Echinococcus granulosus occurs in New York, and that's one that you could get from ingestion. Mites and the carcasses that you might be picking up have all sorts of bacteria on them, salmonella, clostridium. And if you're looking at a large bird kill or another type of animal kill where there might be pesticides involved, you would want to protect yourself from those pesticides that might be on the carcass when you're handling them using PPE so it doesn't absorb into your skin. So the way that these agents typically get into your body are through mucous membranes. They're the first one we talk about. Your eyes are mucous membranes, your nose, and your mouth. And there's a couple other mucous membranes that don't usually come into play during work hours. Ingestion, if you were to get any of these materials into your mouth, could be an exposure. And you're saying, well, I'm not going to lick the carcass. I've got some common sense. I'm not going to get stuff into my mouth. But it's very easy to lose track of what you're doing and then eat something later before washing your hands or taking your gloves off. I see all these folks, not a lot of them, but some people are not only wearing face masks to protect themselves from COVID-19, but they're also wearing gloves, and they're wearing gloves to do everything. And they're really actually probably putting themselves more at risk by doing that than they were if they weren't wearing gloves, because they forget why they have them on, and they would expose themselves. Absorption through the skin is typically, a lot of the pesticides are able to go right through your skin and into your bloodstream. So you want to protect your skin from that. And then there's injection. Injection would be if you were picking up a carcass and there was an old broadhead in there that you cut your hand on, or a sharp bone fragment that went through your glove and into your skin. That would be an injection. And certainly an animal bite would be an injection. That's the way that rabies is typically transferred, is through the bite of an animal, which is a classic ingestion. So I've got some PPE here that I'm going to show you. I'm going to demonstrate some of them. We're going to talk about them. And then at the end, if there's time, I'll just demonstrate bagging up a small specimen. So the absolute minimum PPE you folks are going to use in the field when you're collecting animals for wildlife health unit to take a look at, or Cornell, are going to be latex gloves. And these is also the minimum you use if you're doing work with live animals, if you're radio collaring fishers or handling bats. Latex gloves would be the minimum amount of PPE that you're going to use. So we'll talk about those first. So latex gloves not only come in latex, they also come in nitrile. And this is an example of a nitrile glove. Nitrile is a little bit stiffer than latex. It's a little bit thicker as a rule, although both of these gloves come in different mills, different thicknesses. Typically, you want to get the thickest one possible for most of the general work that we're doing. Advantages of nitrile is it's a little bit more puncture resistant. It comes in a lot of different colors. I'm not a huge fan of the black that I have here because it doesn't really show a lot of blood. I like a white glove that easily shows contamination with blood or fluids or soil. When you're using latex gloves, these do expire over time, right? So if you had a box of these in your vehicle for a long time, they deteriorate, and when you go to put them on, they'll tear. So you want to be careful with that. I'm going to show you essentially how to put these gloves on and how to take them off. I don't know if you can see me at this distance. Putting them on doesn't seem to be that much of an issue. If you're wearing a watch, or if you're wearing rings, like a big diamond ring or something that might puncture the glove, you're going to want to take those off. And it's important to choose gloves that fit you properly. If the gloves are too small, every time you go to move your hand, it's like an exercise machine. There's a lot of resistance there. If they're too large, you're going to have the tips of the fingers sticking out. It's going to be difficult to manipulate things, tying knots, certainly working with a scalpel. You could easily cut the tip of your glove off. So when you have these gloves on, I want you to remember that these are to protect your skin from getting contaminated with bacteria, viruses, etc. But you don't want to, when you're using them, once they become soiled, you certainly don't want to get them near any of your mucous membranes. You don't want to scratch your eye. You don't want to pick your nose. You don't want to pick your teeth. You want to be careful about handling pens and paper, because everything, once the glove's contaminated, once you touch a pen or paper, you're contaminating that pen or paper also. So you certainly don't want to put a pen in your hand and then put it in your mouth later. So you want to be cognizant of why you're wearing these gloves. And one way that I kind of tell people that to remind them is to imagine that once you put them on, there's something on here that's really gross that you don't want to get into your body, or something you're really afraid of, right? So Ebola virus is one that no one wants to die from bleeding out their eyes and all their orifices. So that kind of helps them pay attention to what they're doing if they imagine that there's Ebola virus on these gloves. So after you do what you're going to do with the gloves, and you want to also be cognizant that you're not touching door handles on vehicles or the steering wheel with these gloves, because the person that comes after you to use that vehicle has no idea that they're potentially contaminated. Your goal then, when you're done, is to take these gloves off without contaminating your skin with the nasty stuff that's on the outside. So I'm not sure how well you can see. I'm going to lean in a little bit close. The way to do this is to grab the opposite glove near the heel of your palm, and you're going to want to pull that glove off inside out. You don't want to aim it up towards a coworker or anything. You want to aim away from people, kind of aim it down, and you're going to pull that glove off inside out, collect it in your hand, and now this hand is theoretically doesn't have anything on it, and you're going to go under the cuff of the other glove with your finger, and it gets harder as they get sweaty. And you're going to do the same thing with that. You're going to pull this one off inside out. So now what's the outside now was the inside, and it's probably uncontaminated. Then you can take this and you can put it in the bag with your carcass to be bagged up, or you could dispose of it in regulated medical waste trash can. You don't want to leave it on the side of the road or in someone's yard where you're collecting a carcass. The first thing you want to do after you take those gloves off is wash your hands with soap and water. Obviously, you usually aren't going to have soap and water available to you in the field. So the next best thing are these alcohol-based hand gels. And the first thing you want to do, put some of that on there, be pretty generous and liberal, and rub that around. And at your first opportunity, when you come to the first McDonald's or Stewart's or wherever, go into the restroom and wash your hands with soap and water. Soap and water is the most effective way to really clean your hands. Other alternatives in the field, now that we have COVID-19, we've got this new New York State Clean hand sanitizer. This is pretty good. These Purells are about 70% alcohol. This stuff's 75% alcohol, and it's got a nice scent. You want to keep all this stuff out of your mucous membranes, though, because it will sting. And there's also like a hand wipe that you can use that's germicidal. And these are nice because you can scrub soil off at the same time. But at your earliest convenience, once you're done with whatever of these you're using, you want to get soap and water and wash your hands as soon as possible, certainly before you eat. Other kind of gloves that are available that we often use to protect ourselves this is like a rubber dishwashing glove. And these are nice because they're thicker than latex gloves. If you were handling a large carcass like a deer and you had to drag it for some distance, these are nice because they've got abrasion resistance. They're fairly comfortable. They've got a little textured surface on them. Certainly if I was at a bird kill where there was a lot of geese or something, I'd wear a pair of these to help collect all those geese and give me a lot more tear resistance than a regular latex glove would. There's also thicker nitrile gloves that I don't have a set here, but if you were handling suspected carcasses with pesticide on them, that would be a good choice because most pesticides wouldn't be able to go through those. if you're handling something like a porcupine or a live animal that you're afraid might bite you, leather gloves like these are great for that. But you have to bear in mind that if you're handling small animals, certainly like a bat, that you lose almost all tactile sensation with these and some mobility if they're too small. So you don't want to crush or damage the animals that you're dealing with. But this would protect you from some bites. These leather gloves are very difficult to disinfect properly, and that's an issue. Sometimes we use inexpensive ones and dispose of them depending on the situation. And then there's certainly larger, if you're dealing with larger animals, a glove like this would protect you from bites, probably to a coyote size. I don't think a coyote could get through this. It would certainly hurt, but I don't think you could bite through that. Another glove that you might come across if you're doing chronic wasting disease tissue collection are these cut-proof gloves. And we wear these on our non-cutting hand while we're collecting tissues. And this particular glove will protect you from a slice from a scalpel or a knife, but it won't protect you from a poke with a sharp edge. We go right through this. So bear that in mind when you're wearing these. And then we wear latex gloves on top of them to protect them while we're working. So those are most of the gloves that we typically use. There's also a metal chainmail glove that we'll use in some situations. I didn't bring one up here, but they're heavy. They're very uncomfortable to wear, but we were using those for a while with some really hot West Nile cases. We did have a case here in the lab where someone did have an exposure from a cut to West Nile virus, and the outcome was good. They lived, but it was serious for a while. The next thing I'm going to talk about is protection for your eyes, because your eyes are a mucous membrane. I wear glasses, but a lot of you don't, and it's easy to get stuff splashed up into your face and into your eyes. The most inexpensive and readily available stuff we have for field crews working in collecting specimens are these face shields. And this is just a sheet of clear mylar, very lightweight. They're crystal clear. They come pre-packaged. And in order to put one on, I don't know if I can get one on with these headphones on. It goes on like that. It's protecting your eyes, and it's protecting your mouth and your nose a little bit from splashing. Alternatively, there's chemical goggles, like in chemistry from the old days, which are really good for protecting your eyes. They look kind of nerdy, but the new versions out are quite stylish. And I don't think I'm going to be able to get them on with this. So I guess I'll skip it. But these look pretty nice, don't they? I would look good in those. And there's also these type goggles that go right on over your glasses. You can carry these in all the vehicles, and that protects you from the front and from the sides and top a little bit. You want to be very careful. You get something like rabies virus in your eye. That's a very serious exposure. So you're going to want to report that right away. Flush your eyes out with plenty of water immediately, as soon as you can, because it's a very short trip from the mucous membrane of your eye into your brain. So next I'm going to talk about respiratory protection. And with COVID-19, there's a lot of different face masks that are out now. The healthcare workers are wearing N95s, so they're protecting themselves from getting the virus, in addition to protecting others. The general face masks that surgical type masks that are available now are mostly protecting other people from you. So when you wear a mask like this is a kind of a new standard state-issued COVID reusable mask. This is helping to keep some of your vapors, your exhaled breath with moisture and saliva in it, and virus potentially from infecting other people. So this is one option. It's not awesome. This is a little better version, I think, because it's lighter weight and it's more comfortable to wear. Bear in mind that there's an outside and an inside. Typically the outside is colored. In this case it's blue. The inside is almost always typically white. And there's a top and a bottom. The top's got a little wire you can feel in there. And you want, I can't get this on with these things on. But you want to make sure when you put it over your nose that you're pushed down on that wire to help you get to a better seal. You're not going to get a great seal with one of these. Air is still going to go in and out from the sides, but this is certainly better than nothing. And then there's the N95 masks that you hear about that the healthcare workers are using, and we use them here in the lab as well. This is a 3M version. These you need to be fit tested for if you're wearing them. They also have a metal wire on the top. This particular one has an exhalation valve. I don't know if you can see that. These exhalation valves are not good if you're wearing these to keep potential SARS virus from animals that are in your care. So Krysten mentioned we're a little concerned about rehab bats and mink and other mustelids. This would be a poor choice for that because this is designed so when you breathe out, the air just comes straight out. Your nasty breath and germs come straight out this little valve here, so it doesn't steam up your glasses. So if you're going to be using it for rehab or for research in the field when you're handling research animals, you want to wear some that don't have an exhalation valve. And this is the style that we wear in our laboratory. It doesn't have an exhalation valve. You need to be fit tested for it. Bear in mind when you're wearing one of these, it is a little more difficult to breathe, and if you're exerting yourself, you could become fatigued pretty quickly with one of these. So keep that in mind. Other things you're going to want to protect are your clothing, right? So you could, if you don't protect your clothing, you can get parasites or germ agents. The COVID-19 virus isn't really that big of a deal on clothing, but rabbit hemorrhagic disease virus can stay active on clothing for a month or more and still infect another rabbit. So you want to protect your clothing. And the way to do that is there's portable gowns like this one, which is just a plastic version of this. This is a style that we wear here in the lab, and this protects your clothing. Then you can pop one of these on, do whatever messy business you're going to do, and then take it off and dispose of it, and it gets burned. For doing work in bat caves, oftentimes they're wearing Tyvek suits, and this is merely just protecting your clothing from getting contaminated. In that case, with the fungi that causes white nose syndrome, but there could be other things depending on what you're using it for. Bear in mind when you're wearing one of these in the summer, they're very hot, and you could lose a lot of moisture, a lot of sweat weight in short order with one of those. If you're going from site to site, you want to protect your shoes, your footwear. You don't want to bring pathogens from one site to another. For example, if you're doing anything with rabbit hemorrhagic disease, or if you're doing amphibian work, you don't want to bring rhinovirus or chytrid fungus from one place to another. So you want to protect your feet. There's disposable booties like this. A little better option might be these rubber boots, which can be disinfected afterwards between sites with bleach or with Virkon. Another thing to keep in mind is if you're doing anything near the road or during hunting season where folks are out hunting, you're certainly going to want to wear some kind of high visibility color so that cars see you and you don't get hit by a car or accidentally mistaken for a prey animal. This has got a reflective tape on it too for headlights. So Lauren's telling me that time is running short here. I'll wrap it up. Other forms of PPE that I didn't really discuss yet is sunscreen. This is actually hand cream, but I didn't have sunscreen. To protect yourself from sunburn and potential skin cancer. Insect repellent to protect yourself from insect bites, which are a vector for several diseases, including West Nile virus and Lyme disease. And then this is actually a syringe to remind you vaccination is also a form of PPE. You're all vaccinated for rabies. Potentially we'll all soon be vaccinated for COVID-19. And so that's a form of personal protective equipment that you want to keep updated. Very quickly, I'll go over the bagging of a carcass. This is going to be our raccoon. A little longer tail than most raccoons. You're gloved up. at a minimum, to pick up a dead raccoon, I'd want to wear gloves and maybe a face shield. Maybe not. I wouldn't be wearing these headphones. So bear in mind that we don't know what happened to this animal. We don't know what diseases it has. We know that it could potentially have one that could kill us if we're not careful, rabies. But if there's a lot of blood, there could be blood loaded up in the fur of the tail. So you want to be very careful that you don't flip that tail back and forth, because that can unleash that blood right into your face or into someone else's face. So you want to, and the same thing with like deer's ears. They can hold a lot of blood, and they are very snappy when you're trying to handle the deer, and they can spread some blood. You want to grab an appropriate size bag that will contain the carcass and give you enough room to tie a knot in it. And the theory here is very similar to when people pick up their dog's poop when they're at the park. You're going to invert the bag over your hands. You're going to grab the carcass, make sure you got that tail in there so he doesn't get anything on you. So right now the outside of the bag was the inside, and it's clean. Tie a simple overhand knot. Other options would be a zip tie or a duct tape, but this is cheaper and it's better. Then you're going to want to remove your gloves. So I got the cuff on this one. And you can put those right on the carcass, and you're going to double bag it so those gloves will come with the carcass for disposal at the lab. And do the same thing with the second bag. Very simple, very easy. And you're good to go. And the first thing you want to do after you do that is plenty of hand sanitizer. So I hope you enjoyed this PPE demonstration. And if anyone has any questions.
Kevin, you have a number of questions.
Oh, awesome.
Are you ready?
Yeah, I'm ready.
All right. So the first one is, can cut-proof gloves be washed and disinfected, or would washing break down the fibers?
We routinely soak these in disinfectant when they become visibly soiled, or at the end of every shift, they get soaked in disinfectant overnight and rinsed out. And they've lasted quite a bit. We're just doing that. We're not putting them in an autoclave. We're putting them in a washing machine. So I'm not sure how they would hold up to that, but I think probably pretty good. It'd probably be a good idea to replace these every season or so. They're not terribly expensive.
All right. The next question was, any suggestions to mitigate fogging of glasses when wearing a mask?
So the best thing to do for glasses fogging, if you can't wear a mask that has an exhalation valve, which is supposed to help for that, and you wouldn't want to wear one of those in the situations that I described, is to fiddle around with that wire at the top and really try to push that down and get a good seal. And it is an issue for people that wear glasses, because sometimes, depending on the respirator, your glasses might be sitting on top of it, depending on where it comes up on your nose. So you really kind of want to try, if you have them available, try a couple different ones out to see which ones best fit your face for your glasses situation, because all glasses are a little bit different too. They do make some stuff that you can put on your glasses that are for anti-fogging. I've never used them, so I'm not quite sure how it works. So yeah, that's the best advice I have for that.
Thank you. Another question, how do gowns or coveralls have a role in preventing reverse zoonoses, if at all?
They wouldn't well, they could. It depends on what let's talk about COVID-19, which is the one we're kind of concerned with now. They wouldn't have a terribly huge role in wearing gowns. The main thing you want to be protecting the animals from in that case is your breath and your respiration, so you're not breathing them into them, and your hands, which you might have coughed into or have virus on them. So gloves and a face mask would be the most important thing there, as far as actually having the virus go from a person to the animal.
Thank you. Another question, can you speak to the availability of N95 respirators these days?
They are still in fairly short supply. We're having trouble getting them here at the lab. We've actually taken some old stock that was purchased back in 2005 that didn't get used then and is now essentially expired. And we took the straps off them because they kept breaking because they were so old. And we installed new straps on a number of boxes of those so that we could use them because they are in short supply. And there might be some ethical considerations because healthcare workers need them currently. So we're in this gray area currently where supply is down and it might be ethically challenging to use them until supply picks back up. But that should probably happen in the next couple of months. All the factories have been ramped up with production, but we haven't seen an easing up on the supply yet at our end.
All right. And then the last couple questions were, one was related to back at the gloves. How are chainmail gloves good against West Nile specifically? I would assume protection against sharp beak claws or talons.
So the main thing with the chainmail gloves back then was that happened right after we had the fellow get exposed, and he had cut himself across the palm. And these chainmail gloves are virtually impervious to cutting with a metal knife. So that was the main thing. They're considered stronger than these, but same deal with these. A scalpel could poke in between them. So it was a measure that gave us some more protection. Certainly was protecting us against slashes, but it wasn't a perfect solution.
All right, and then going to the bagging of carcasses. There's a question about the air squeezed out of the bag. Any concern regarding disease transmission?
Well, yeah, you certainly want to squeeze the air out of the bag, like into your face or someone else's face. I'm trying to think back on how I did it just now, whether I did that or not. I don't think I did. It's gross. Probably not a huge threat. I'm going through all the diseases in my head trying to think of one that would be a problem. Anthrax might be an issue with that, but I don't think that's a huge issue.
And then what about the pathology tag? At what step should that be included in the bagging of the carcass?
After you wash your hands with the Purell, then you can handle a pen and a pathology tag if you're using an old style pathology tag. A lot of the tags are done probably back at the office using a computer, then they print that out. It is a good situation to have that tag like in between the two bags so it doesn't get lost. So in the case like this, I would tie it on there really well. If I'm using the old tag, fold it up, put it in a Ziploc bag and slide it in between another bag and give it an actual third bag. We don't have a problem with using too many bags. We do have problems with bags leaking because bones poke out of them, and then if carcasses are frozen, leakage really becomes a problem. So triple bagging it isn't a bad idea. So you can put that in between the second and third bag to keep it with the carcass.
All right, and the last thing is just a note about the N95 expiration dates. Keith writes in, the date is because the elastic strap degrades and the filter mask itself is still fine. So,
and that was our experience. So I tried to put this one on earlier today, and these little blue straps, I mean, they just snapped because they were 20 something years old. So we went to the craft store and got some elastic, and Lauren re-outfitted them all for us. And this works pretty well. And I can feel I've got a good fit when this is on. So we're confident that these are going to work for us. It's not an ideal situation, but it's a situation that we're in.
all right. Well, thank you, Kevin. Appreciate that. I'll turn the controls over to Dr. Brenda Hanley, who is a mathematician, and she's going to talk about some of the modeling work that's being done around COVID to help us understand it more. So.
Okay, let me share my screen here. Okay, can you all see my screen now?
Yes.
Okay, great. All right. Okay, so like Krysten said, my name is Brenda Hanley, and my specialty is in mathematics, statistics, and computational biology. I'm a postdoc here in the Cornell Wildlife Health Lab. And although I am not a COVID modeler, I am a mathematical modeler, and I regularly use and develop models, the same class of models that are being used to model COVID-19. Now, my goal with this presentation is to give you a crash course in mathematical modeling and to put into perspective some of the topics that you've heard regarding mathematical modeling in the news. Okay, so models in general are tools that we develop and use to facilitate the investigation of some natural process. We also use mathematical models to understand what we need to do in order to change the trajectory of some natural process. So let's go through a very quick example of a type of model, and we'll talk about the different components that make up a mathematical model. So say we wanted to go on a hike, and we went up and we found some beautiful lunch vista. And we wanted to infer what elevation we were sitting at, but we didn't have a GPS, a topo map, and there was no sign up there saying what elevation we could. We could use the tools at our disposal to infer the elevation of our lunch vista using a mathematical model. So in this case, the mathematical model is a model for a line that you might be familiar with from algebra. So here we have y equals mx plus b. Our variables, or the things that we would like to infer relationships between, are going to be x, the time we spent walking, and y, our ultimate lunch elevation. In that model, we also have two coefficients. The coefficients b is going to be your intercept, or your starting elevation at the trailhead, and m is going to be the slope that you walked up on your hike. Now, we can think about this in terms of our starting elevation. We can kind of know that it's going to take a lot longer to get to, say, the top of Mount Everest if we start at sea level versus if we start at base camp. Similarly, m also has an influence on x and y. If we walk a trail that has been developed for stock use, we know that it's got a relatively small slope, versus if we were to walk a game trail, we'd go right up the ridge and we get to the summit in a hurry. So ultimately what we have is we have a mathematical skeleton, which is the model, but that relationship between the two things that we want to know about are also scaled by the coefficients in that. So we regularly use mathematical models in wildlife management. If we want to depict how an exponential growth of a population, we use a class of models called exponential models. If we want to investigate exponential growth up to some carrying capacity, we can use logistic models. If we want to add demographic structure into those models, we can use population matrix models. Or if we want to depict how an infectious disease moves through a population, we can use a class of models called SIR models. So the movement of COVID-19 through the global population of humans has been modeled using an SIR model framework. There are other types of models called agent-based models, and I'm not going to go into them in this presentation, but you're more than welcome to ask me about agent-based models at a later time. But ultimately, let's talk about some SIR modeling framework. So SIR stands for susceptible, infective, and recovered and or removed. In the case of COVID-19, we have a recovery and we also have removed. So it's adaptable to the pathogen that we're trying to model. And ultimately, we can see that we have the variables and we also have coefficients in this model. The variables are the compartment of susceptible individuals, compartment of infective individuals, and the compartment of recovered individuals. And then we have the coefficients, which is the transfer of individuals among those compartments, which is represented by the blue. Now we have a few assumptions of this class of SIR model. New susceptible individuals enter the system through birth, or in the case of an emerging disease like with COVID-19, new susceptibles are in the system as susceptible simply by existing on the planet when the pathogen emerged. Each person is in one compartment at any given moment, so this can be thought of as an accounting system of where everybody is in their state of their disease at any given time. That being said, the number of susceptibles, the number of infectives, and the number of recovered adds up to the total population size. As well, in this particular type of model, the process is irreversible. So you can see there's the directionality of the coefficients in blue. Now, in an SIR framework, you can also add complications to better represent the pathogen that you're trying to study. So for instance, you have an SEIR model, which contains a latent or exposed state. In this capacity, you would have an exposed compartment that falls between the susceptible and infective compartments. You can also have an SIRS model, which allows recovered individuals to again regain susceptibility. So in this case, we would have a fourth compartment that's to the right of the recovered compartment, and it would start the cycle all over again. Or you can have both types of complications, an SEIRS, where you have an exposed state and recovered individuals become susceptible again. So let's look at the mathematics of these coefficients. So again, these coefficients allow for the transfer of matter between these specific compartments. So B is your transmission coefficient, or in this case, beta. That is actually equal to the probability of a contact among an infective individual and a susceptible individual times the probability that that infective person will successfully transmit the pathogen to a susceptible individual. You also have some probabilities of removal by natural death. Now, remember, we have individuals, and individuals are moving through their life cycle in addition to the pathogen moving through the population. So that's going to be your mu. And then you also have the recovery rate. So this gamma is the probability that an infective individual will enter the recovered stage or the removed stage. Now this model, this depiction of the model can be collapsed into the mathematical equations here. So what we have is we have a whole bunch of variables, in this case the counts of susceptible and infective individuals, and then we have them scaled by some coefficient values, which is your beta, your mu, and your gamma. What these say is that the rate of change of susceptibles is going to be proportional to the amount of susceptibles that leave the susceptible category and enter the infective category, minus the number of susceptibles that leave the susceptible category as a result of natural death. Similarly, you have the rate of change of the infective category is going to be proportional to the number of susceptibles that then become infective, minus the number of previously infective that recover or are removed, and then minus off the number of infectives that are removed from the system for natural death. Now again, we have this mathematical framework, but we also have some coefficients. And those coefficients, just like in our hiking example, are going to scale the magnitude of that outbreak over and across time. Now we would like to know the actual values for all of these representations that I have, but sometimes we don't know those values, but we can still analyze the actual mathematical model in a theoretical standpoint in order to understand some basic thresholds about the outbreak. One of those theoretical thresholds is called the RO. You might have heard this in the news lately. It's also called the reproductive number. The RO is a basic mathematical threshold that says under the conditions that an outbreak will occur and under the conditions that the threat of an outbreak will subside. If our reproductive number is greater than 1, an outbreak will occur. What that means is that if a single infected individual is infecting more than just replacement value, more than just a single other person. So if RO is equal to 1.1 or 1.2, for instance, that means that the number of cases are growing. If the single infective individual does not replace itself in terms of an open case, then the threat of the outbreak will subside. So something that you may have heard about in some of the press conferences is talking about an RO of 1.1 versus 0.9. If you have 1.1, that means that you're going to have very likely that an outbreak will occur. If you have it at 0.9, it will eventually subside. Now notice, this is a function of both your population size, which is N, which again is the sum of your number of susceptibles, infective, and recovers. And it's scaled by those transmission coefficients, or your beta and your gamma. So again, the reason why we're so interested in testing is to understand what the values of those coefficients are. Now, here is an actual SEIR model that has been used to represent COVID-19. This, you should see some of the variables that we have. We still have a susceptible compartment, an exposed compartment. This time we have 4 different types of infective compartments, and then we have a bunch of different ways that you could remove yourself from the system, or that you can be removed from the system. Notice the transfers among all of these different variables are coefficients, the blue. All of these are numbers, and all of those numbers need to be measured through testing. Now, when we have a pathogen that is moving through a system, the pathogen is not only dictating the structure of the model or the different configurations of the orange components that we have, but it's also dictating the values of those coefficients. And so we have a huge number of moving parts. And so when we model this, we can understand a lot theoretically about it, but a lot of the reason why we're modeling it is not just to understand it, it's also to understand what we do about it. So when you hear in the news, like, it depends, what do we do, it depends on a lot of things. The first and foremost, it depends on the structure of the model and how it moves through the system. And that is directly dictated by the functionality of the pathogen itself. But the optimal strategies that we choose to employ also depends on the strategies that are palatable to society. Now, the problem with COVID-19 is that this emerged and rapidly started spreading around the world. Every single human on the globe immediately found themselves in the susceptible category. And so even if we didn't know all the values of all of these different coefficients, the most obvious single point failure in this system is to just halt transmission between the susceptible and the exposed categories. So this was the motivation behind the stay-at-home orders that happened very early. If we can decrease that beta value, then it doesn't matter what happens downstream, because we could potentially stop this outbreak. So let's look at some of the data for the US. So here, this is a graph that I've put together of the daily growth rate of open COVID-19 cases in the entire United States. This is publicly available data that's taken from the covidtracking.com. And what this shows is the open number of cases in the United States as a whole per day. And so on the x-axis, we have the daily growth rate of open cases. And on the y-axis, on the y-axis, we have daily growth rate of open cases. And on the x-axis, we have the calendar date in 2020. The red line is the growth rate of 1, which corresponds to the plateau. That growth rate means that the number of newly infected people is equal to the number of people that are recovered from the disease or being removed by the disease. That corresponds to the plateau of the curve, or the top of the curve, when the number coming in is balanced with the number going out. The purple line at this daily growth rate of 2 indicates a doubling time. And the green line indicates the complete extirpation of the disease where there are no open cases. The blue is the time series data of open cases from publicly available data in the United States. Notice in early March, we had a daily growth rate of around 1.38. What that means is that if we have 100 open cases today and a growth rate of 1.38, that means that we have 138 open cases tomorrow. If we have 1,000 open cases today, a growth rate of 1.38 means that we have 1,380 open cases tomorrow. So you can see at the beginning of March, when we had unabated growth of open COVID-19 cases, we were adding cases at quite literally an exponential rate. Now, trying to put together shelter-in-place orders all over the country, you can see at the end of March, we started changing the transmission of this pathogen through our communities. And so you can see that blue line started decreasing. What we did was we essentially changed our behavior to cut off transmission among open cases and the remaining susceptible cases. And so as you can see, those shelter-in-place orders were helping, and that blue line tended towards that red line. Now, in New Zealand, for instance, that blue line had crossed the green line. They had successfully extirpated COVID-19 in their entire country, up until sadly last night when I read a news article that there are now 2 open cases. But extirpation of the disease is when that blue line crosses the green line. We right now are at that red line. That red line is the plateau. That red line is the top of the curve, which says that we have the same number of new cases as we have people recovering or being removed. So the most recent numbers that I'm seeing is that we are adding 20,000 new cases a day, which indicates that we are recovering 20,000 or we are removing 20,000 per day. So that's what the mathematics says. Ideally, we would like that blue line to be below 1, which means that the outbreak will eventually subside. But right now we are at that red line, which means that we are still at the top of the curve. Now, I know that a lot of questions remain, not only in the mathematical modeling world, but also in the world of interpretation of the mathematical modeling. So if you guys have any math questions related to what you guys see, you're more than welcome to email me, and I can try to translate what's going on deep in the differential equations to any questions that you might have. Okay.
All right. Are there any questions for Brenda? All right. I see thank you, Brenda. That was really well explained.
You're welcome.
Well, Brenda shared her email address, so if you want to follow up with her, if you have any questions. Are there any other questions about the workshop or anything that you've heard today? If you want to type it into the Q&A, we'll wait a little bit. If you want to go ahead and email any of the 3 of us, we're happy to answer those and to get back to you. Once we have the recording, I'll put up on the website with resources. We will send that out to you so you can watch it again or make your friends and family watch it. So thank you all for joining us today. We really appreciate your time and attention. We'll be doing these virtual wildlife health workshops every month. I believe it's the 3rd Wednesday of the month, same time. So go ahead and get registered for those for the next one, and we'll look forward to doing this again. So thank you. Appreciate you being here.